Hi everyone, thank you so much for taking the time to watch this video. This video will discuss the idea of rats as models for opioid abuse for humans by Kira and myself, Zaid.
To delve a bit deeper into this subject, we will be asking ourselves, why can we use rat brain research for human applications when it comes to physical addiction of opioid use? If you didn't know, opioid abuse among humans has become a very hot topic among scientists due to the increase in overdoses and prescription rates. In most research, rats are used as a model for humans, especially for discovery of new medications. But why is that, and how accurate is this assumption that humans are so similar to rats? Before we begin, let me give you some broad information about opioids. Opioids are made from opium, which are derived from poppy flower seeds. The use of opioids has been around for thousands of years in order to cause euphoria and decrease pain. It most commonly seen in doctors' offices as morphine and illegally as heroin. We can see in the photo that in both mice and humans, the opioid receptors inside the body respond to natural endorphins in order to relieve a chemical called dopamine. Dopamine is the chemical that gives us the sense of euphoria. When the body has unnatural opioids or synthetic opioids, it binds to the opioid receptor to increase the amount of dopamine released. While this can be beneficial during surgery to avoid pain, the sensations can be highly addictive. More about the specific mechanism will be discussed later on.
Here we have the mu opioid receptor. These receptors are one of the three major types of opiate receptors, the other two being delta and kappa opiate receptors. The reason why we are focusing on the mu receptor is due to its direct association and link to opiate dependence and reward. As we can see in this diagram provided, there are multiple components that comprise the overall structure of the mu receptor, as the mu receptor is an inhibitory G-coupled receptor. There is a very high affinity that's present between mu receptors and opiates. So in this case, when we see morphine introduced, it will bind to the surface of the receptor, which will cause the alpha subunit, the yellow round circle in the diagram, to undergo a conformational change that will halt the production of cyclic AMP, which will in turn close the sodium and potassium channels. Upon the closure of these channels, the release of neurotransmitter GABA will be inhibited, and which will directly result in the increased release of dopamine.
Think of the mechanism like a pile of dominoes. After each step, one starts slowly falling down, starting with opioid binding and eventually leading to the increase of dopamine.
So now, having taken a look at mu receptors in terms of their structures and mechanism of activation, we can now take a look at how they compare in rats and humans. And taking a look at this research article figure here, we can see that yes, there are a lot of letters, but these letters are in fact genetic sequences, and these genetic sequences make up mu receptors in humans, rats, and mice. And looking at these sequences, we can see that they are in fact very similar, and that there are in fact portions that are identical. And what this just supports is the idea that mu receptors in rodents and humans are relatively the same in terms of their structure and function. And this just shows how rats could potentially serve as really useful and helpful models in understanding the role that mu receptors play in addiction and physical dependence.
While this is very interesting, we can also see other research done by Dr. Brigitte Kiefer, the scientist recognized for finding the genetic link between mu receptors. In her speech at the National Institute of Health, She presents the photo on the right and explains that while the mu receptors are genetically similar, the placement of the receptors are different in rat brains than in human brains. This difference could be the reason for the discrepancies in studies about opioid abuse in humans. For example, in the rat park study conducted by Dr. Bruce K. Alexander, rats who have been exposed to sweetened morphine for 57 days, when given the opportunity to drink water, chose the water, even though they experienced slight withdrawal. This is not something we see happening in humans. Many times they'll socially isolate themselves and they would prefer morphine over anything. We can conclude that while rats are a very good physical model for opioid addiction, they are not perfect. There are still some physiological and behavioral factors that have not fully been explored that might be able to provide better answers as to the relationship between opioid addiction and the physical dependency in humans. The location of the mu receptors in the rat brain differs slightly in comparison to the human brain, but we as scientists have come close to finding a solution for these differences. While studies begin on rats, they are extended to human trials in a safe and humane way, where ethics are taken into consideration. These human trials are vital in finding the answers to our questions about opioid addiction.